plant tissue culture. First, we are going to see about what is the process which is involved in the plant tissue culture processing. First one is intact the plants, then excision of explant, then proper trimming of explant, then next go for a surface sterilization of explant, then what are the materials which is going to involve in the P6 chamber. That material should be sterilized with their chemicals, that is sterilization process, that is sterilization of glasswares, and also we are sterilized with their media and their utilities. The explant washed in sterile distilled water and culture medium is sterilized in the autoclave. Then it is inoculation of explant in the culture medium under the aseptic conditions, incubation under the suitable conditions for a proper culture. Then it go for a subculturing for obtaining the desired results. Then go for a planted transformation. These are the steps which is involved in the plant tissue culture program. These are the steps in general technique for the plant tissue culture program. First, we are seeing the one plant. This plant regenerates the regeneration pathway, that is explant, which is go for a direct organogenesis process, then direct somatic embryogenesis process. These explants going for a callus formation. In this callus formation, we are go for a somatic embryo and then direct organogenesis, that is root organogenesis, then shoot organogenesis. In this shoot organogenesis, we are go for a plantlet and then indirect somatic embryogenesis. This is a plant regeneration pathway. These are the some examples for the plant tissue culture plants which is grow under their aseptic condition, that is plant tissue culture lab. We are going to grown. Okay, these are the plants which is grow under their bottles. The next one is the explants are inoculated in a PTC chamber. Introduction. The plant tissue culture has a great significance in the plant biotechnological, especially in the crop improvement programs. The term plant tissue culture may be defined as the process of in vitro of explants, that is, pieces of living differentiated tissues in the nutrition medium under the aseptic conditions. And the tissue culture includes the term tissue culture as well as cell culture, organ culture and suspension culture also. Thus, the tissue culture has become a great interest of molecular biologists, plant breeders and even though industrials, it helps in the improvement of plants of economic importance. Then, in addition to this, to all this, the tissue culture contributes immensely for understanding the patterns and responsible factors for growth, metabolism, morphogenesis and differentiation of plants. Tissue culture. The in vitro culture of tissues, that is a callus culture. Cell culture denotes the in vitro culture of single or a few cells, that is called as a cell culture. Organ culture. The term is used for the in vitro culturing of organ like embryo, root and zooapex. What is the suspension culture? Defined as a culture of cell and cell aggregate which is going suspended in a liquid medium is called as a suspension. That is, the cell or cell aggregate which is suspended in a medium is called as a suspension culture. Then explant. What is the explant? Explant is called as the excision of piece. That is what plant material which is going to inoculate. That is the excision of piece, which is excision of piece of differentiated tissues are the organ which is used to culture is called as a explant. That is excision of piece of differentiated tissue or the organ which is used for a culturing is called as the explant. That is embryo, young leaf or bud, etc. What is a callus? Callus is an undifferentiated mass of cells is called as a callus. The undifferentiated mass of cells. That is these cells are we are going to differentiate. The callus is a mass of cells. That is what is a callus means? That is a mass of cells. That is undifferentiated mass of cells is referred to as a callus. The cells of callus are meristematic in nature. History of plant tissue culture. The first work in plant, the Haberland and German botanist G. Haberland as a German botanist in the year of 1902 cultured a fully differentiated plant cells isolated from the different plants. This is a very few steps for the beginning of the plant cell and tissue culture. Further contributions were made by the cell doctrine which has admitted that a cell is capable of showing a totipotency. With the identification of a variety of chemicals like a cytosine, auxin and other hormones, vitamins etc. and their role in the affecting of cell division and differentiation, the methods of plant tissue culture developing in a proper manner. The three other scientists, Gothard, White and Norbercote, also made a valuable contribution to the development of plant tissue culture techniques. Later on, a number of suitable culture media were developed for the culturing of plant cells, tissues, protoplast, embryos, and thus root tips. For example, we are culturing like a protoplast. We are choosing for the protoplast medium. So we are going to inoculate the cells or tissues which is chosen for the separate media. These are the later on a number of suitable media were developed. The discovery and understanding of role of plant growth hormones in the multiplication of cell also provided an extra aids for the development of in vitro culture methods of plants. The first plant from the mature plant cell was regenerated by the Baron in the year of 1959. Foundation of commercial plant tissue culture was aided in the year of 1960 with the discovery of a million of fold increase in the multiplication of Crymborium, which was accomplished by the Chiomoril. In India, the work on tissue culture was initiated during the year of initiated during year of 1950s at the University of Delhi. This initiation is credited by the Star Panshan Mageshwari, who was working there in the Department of Botany. Discovery of haploid production was a landmark in the development of in vitro culturing of plants. Sri S.C. Mageshwari and Sripa Guha made a remarkable contribution in the development of plant tissue culture in India. Later on, the development in this composition of nutrient media and genetic engineering served as a basics for the further success in the plant tissue culture techniques. Then Haberland was the first person who made attempts in plant tissue culture. That is, he developed the concepts of in vitro culture of plants, and it is acceptably regarded as a father of tissue culture. So these are some achievements which is displayed in the tabular column. In the year of 1902, we already say that Haberland, the first attempt was of in vitro culturing of plant cell, and then 1902 hanging, 1922 Robbins in the year of in vitro culturing of fruits. In the year of 1925 Leibich zygotic embryo culture in the Unium. In the year of 1934 White culturing of fruits of tomato plant. In the year of 1939 Godre and White and Nobukood successful establishment of infinite callus culture. Then in the year of 1941 Baron culturing of drone gall diseases. In the year of 1948 Lou culture from the stem strip. In the year of 1955 Miller hormone chitin discovered. Then in the year of 1957, Skoog and Miller discovered the axon-cytokine ratio regulates their organ formation. In the year of 1960, Beckman development of planting techniques for the isolation of single cell. In the year of 1970, Power, the successful protoplast fusion. The 1970, Mageshwari and Gupta successful anther culture. In the year of 1974, Richard Lynchhand bioformation in the plant tissue culture. In the year of 1978, Marlich's production of somatic embryo tomatoes. Morphogenesis. What is a morphogenesis? The shaping of an organism by embryological process of differentiation of cells, tissues and organs and the development of organ system according to their genetic. That is a blueprint of the potential organism and environmental conditions. What are the differentiation process which is in plant? Differentiation is the process of the cells of the root or shoot, apical meristem and cambium matured to perform a specific function. This is a process. A lot of structural changes occurs in the plant cells. What is this potency? Cellular totipotency. What is the potency of plant cell to grow and develop into a whole new multicellular plant is described as a cellular totipotency. What is a totipotency means? In other words, the property of a single cell for differentiating into many other cell types is called as a totipotency. This is the property which is found only in living plant cells and not in animals. This property only having a plant cell, not in animal cells, excepting this being stem cells in animals. The term totipotency was coined in the year of 1901 by Morgan. During culture practice, an explant is taken from a differentiated mature tissue. It means the cell in the explants are generally non-dividing and quiescent in nature. To show the totipotency, such mature non-dividing cell undergo the changes which revert them into a meristematic state, that is usually a cellular state. This phenomenon of reverting a back of mature cells to dividing a state is called as a de-differentiation stage. Now these de-differentiation cells has to be ability to form a whole plant or plant organ. This is a phenomena is termed as a de-differentiation. That is the explant, mature, non-dividing and differentiated cells. That is differentiation process. The callus, that is undifferentiated and meristematic cell, that is go for a redifferentiation process. That is new plants and dedifferentiation, etc. The de-differentiation and redifferentiation are the two inherent phenomena involved in the cellular totipotency. Regarding this, it is clear that the cell differentiation is the basic event for development of plant, and it is also referred to as act as a cytodifferentiation. For example, this picture shows a mature plant is there in a pot. This plant leaf, that is for a mature leaf washed with a disinfectant for sterilization process. Go for a sterilization process in a leaf. Then a small pieces which will be cutted. A piece of cut from a leaf will be cut. These cells are isolated with homogenizer and cultured. The leaf cells were singled. Then cell division process which goes, the callus will be formed. Then this callus will be formation of shoot buds. In the shoot buds isolated and washed and subcultured, then plantlets will be grown, then again for a mature differentiation. Applications of totipotency. Various applications of cellular totipotency are: it has potential applications in the crop plant improvement, micropropagation of commercially important plants, the production of artificial and synthetic seeds. It helps in the conservation of germplasm, that is genetic resources, in this ability to utilize for the haploid productions. Applied in the producing the somatic hybrids and hybrids helps in the cultivation of those plants whose seeds are very minute and difficult to germinate. Also helps to study the cytological and histological differentiation for higher scale and efficient production of secondary metabolites. The genotypic modification can also be possible. Plant tissue culture laboratory. The cell or tissue are obtained from any part of plant like stem, root, leaf, etc., which are encouraged to produce a more cell in a culture and to express their totipotency, that is their genetic ability to produce more plants. Cell or tissue are grown in different types of glass via containing a medium with mineral, nutrients, vitamins and phytochromes. Therefore, to carry out the experiment using the tissue culture techniques as well equipped laboratory is the first required. An ideal tissue culture laboratory should have at least 2 big rooms and a small room. One big room is for a general laboratory work such as a preparation of media, autoclaving, distillation of water, etc. The other big room is for keeping a cultures under the controlled light, temperature and humidity. The small room is for aseptic work for keeping autoclave articles. General laboratory, a washing area. This is a very important for a tissue culture laboratory. It should be provided with a large sink, running hot and cold tap waters, brushes of various sizes, detergent and bucket for a single distilled water for a final rinsing of the water. Glass hoods. Hot air remedy. It is necessary for drying the washed glasses good. For this purpose, a number of enable drying of different sizes are required for keeping a wet glasses good inside the oven. Refrigerator is essential for storing various thermolable chemicals like vitamins, hormones, amino acids, casein hydrolytes, yeast extract, then coconut milk, etc. The circulation of salts were also kept in the prevent contaminations. Distillation plant, which is very important. A single distillation and double distillation water plant were indispensable. 2 big plastic containers are required for the distilled water. Weighing balance. 3 types of weighing balance via pan balance, chemical balance and electronic balance are required for the weighing chemicals, sugars, agar-agar and others. They are also needed for the pH meter, vacuum pump, autoclave. pH meter to check the pH values, vacuum pump which is maintain the vacuum. That is it is required for the filtering of liquid media, sugar solutions through the filter apparatus using the as liquid. Autoclave is very important for sterilization of glassware and glass goods. Working tables, it's necessary for a preparation of medium. Heater, need for the heating. Microscope is a simple compound and inverted binocular microscope which is used to observe their different objects. Microtome also which is needed. Wooden racks, laboratory for aseptic inoculations. Culture room, which is very important. The culture room means the room for keeping incubating the culture under the controlled temperature, light, humidity. The culture room is also filtered with the double doors in order to make it dust free and maintain the constant temperature. Thank you.